Our next speaker is Tania Sorg, who will be talking about a new mouse model for Costello syndrome.
So good morning, and thanks a lot to the organizer for giving me this opportunity to present here our work about the generation and characterization of a new mouse model for the Costello syndrome. So the Costello syndrome has been first described in 1971 by Dr. Costello. It's a rare disease and is revealed right at birth, almost at birth, or at least in the first months of life. And there are about only 300 cases that are reported worldwide. It's a faciocutaneoskeletal syndrome, and the principal characteristics are postnatal growth delay, facial features, as you can see on these pictures, redundant skin, laxity of the joints, tight Achilles tendons, and as well as a development delay and mental retardation. But there are also some associated malformations that are usually the cause of the death, such as the cardiac abnormalities with hypertrophy or tachycardia, but also some neurological abnormalities. And some of these children have also a high risk of tumor developments in about 15% of the cases, which is mainly rhabdomyosarcoma. So far, there is unfortunately no treatment for these patients, and the prognosis is dependent on the severity of the cardiomyopathy and the tumors. So the Costello syndrome is a rhizopathy, which is a big family of rare diseases. And in this family, we have also, for example, the Noonan syndrome, where we have a mutation of the CHIP2 gene in about 30% of the cases, as well as the Leopard syndrome, also with mutation in CHIP2. And here, for the Costello syndrome, we have a mutation in the HRAS gene. This mutation is often sporadic, and in 90% of the case, it's a de novo mutation. And it leads to a permanent activation of the HRAS protein, leading ultimately to cell overgrowth and abnormal cell division. It's autosomal dominant, so the patients are all heterozygous, and there is a very low risk of recurrence. And there are a lot of mutations in the HRAS gene, and the most common in this Costello syndrome is the glycine to serine mutation in position 12. So we have introduced this mutation in the mouse HRAS genes. We obtained the cells from Mario Barbaseed containing the wild-type HRAS, and we introduced the G12S point mutation by homologous recombination. We established then both homozygous and heterozygous colonies to go through an extended phenotyping process covering metabolism, cardiac exploration, behavior, and cognition. So I will here show you some of these results, but that's more than 2 years of study, so these are the main results that we observed. First, what about the general health and basic sensory motor function of these mice? You can see that in the Schirper assay, these mice are almost inactive. They react abnormally upon transfer. They have also some reduced motor coordination in the rotor rod, so they are falling from the rod faster than the wild-type mice. And they have also a reduced muscular strength in the grip test, which is going along with the human situation. We also evaluated the behavior of these mice in an open field to record not only the locomotor activity, but also the reaction in a novel environment and the evaluation of the anxiety with the center of the arena leading to anxiety if such a phenotype is present. And what we can see on the left is that the total distance traveled in this arena over 30 minutes is reduced in the HRAS mutant. There is also a tendency to a reduction in the number of rears, but there is no difference in the percentage of time in the center of the arena between the mutants and the wild types. So again, we can see in this assay that there is a reduced locomotor activity, while the percentage of the time spent in the center is similar. So these mice are less active, but there is no effect of the HRAS mutation on the anxiety level. We also evaluated the gait in these mice, and this has been performed on the Gait, which measured about 200 parameters when the mouse is working, such as the stride length, the stance, so when the mouse is moving the paw and then landing, and what is also the distance, the angles, and the frequency, and so on. And what we observed, and here I have both the heterozygous and the homozygous, is that in comparison to the wild-type mice that are in each graph on the left, there is an increase in the swing, the stance, the stride, and the break, as well as the proper duration. And both, this can be also measured and distinguished between the fore and the hind limbs. And we have also an increase in the stride length, and the frequency is lower. So it's like these mice are doing big steps, but at a lower frequency. And as we have some defects in the locomotor activity and also in the gait analysis, we also evaluated by electromyogram what is happening in the muscle of these mice. And indeed, there is an increase, and here again, both in heterozygous and homozygous mice, of the SCNV, as well as a decrease of the CMAP in the gastrocnemius muscle, and an increase H-lentil-C in the plantar muscle. So there is clearly a defect in the muscle. We also evaluated by histology if there is a defect in the myelinization, which is not the case, and we did not observe any defect in the in vitro muscular contractility. Because the patients have also intellectual disabilities, we also performed a number of learning and memory assays in these mice. And here we have the novel object recognition. And what we can see in the first graph on the left is that the recognition index is reduced in these mutant mice, both heterozygous and homozygous. And in the graph that is in the middle, you can see that the wild types prefer a new object and do not go back to the familiar object that they already saw the day before or a few hours before. While for the mutants, there is almost no discrimination. There is a tendency, but it's not statistically significant. And we can also see that these mutants have abnormalities in the PPI. So there is clearly also in these mutants some learning and memory phenotypes. What about the cardiac function, which has a big impact in patients? What we observed is that these mice have an increase of the systolic blood pressure that has been measured by a non-invasive blood pressure measurement by takeoff. And they have also an increased rhythm and the tachycardia. So there is a hypertensive phenotype associated to tachycardia in these mice. So we then went through a telemetric recording over a complete day-night cycle. And we measured the blood pressure in various situations, and this is still ongoing, either at the baseline levels or by challenging the mice with a low sodium diet. And what is also ongoing is a challenge with losartan, as it has been described that this mechanism goes through the angiotensin II receptors. And what we observed here is that by calculating the blood pressure, the blood pressure pulse, sorry, I forgot the word pulse. So the pulse pressure represents the difference between the systolic and the diastolic functions, and usually this is something that is happening when the arterial walls are aging. And this is reflecting a defect in the aorta compliance. So these mice in red and in pink have an increased pulse pressure. That means that there is a loss of the artery compliance and a higher rigidity in comparison to the wild-type mice. And this is leading to the hypertension phenotype. And also in echocardiography, we can see that there is an increase in the pulmonary injection time, as well as an increase in the aortic cardiac output, and also a slight left ventricular hypertrophy on the right side. So there is a diastolic dysfunction as well, that results in some abnormalities in the myocardial reaction, and that suggests probably a pulmonary valve stenosis, but we do not have yet the morphological proof of this one. And this is also something that is happening very early in the children that have Costello syndrome. So it's probably linked to already an embryologic step and some defects in the neural crest that we'll also analyze. And finally, we performed some micro-CT imaging. From an external point of view, the gross examination did not reveal any facial dysmorphia in these mice. But by micro-CT, we can see, and the red is the mean of a normal head, or a head, I would say. We can see on the right side that these mice have a macrocephaly, which is also related to what can be observed in human patients. So in summary, if we compare the human clinic and the mouse model that we have generated here with the G12S mutation, we have a number of symptoms that can be reproduced in our mouse model, which is the facial features or the macrocephaly, cardiac abnormalities with hypertension and also some defect in the cardiac rhythm, reduced muscular strength, reduced locomotor activity, as well as a reduced locomotor coordination. And there are also some abnormalities in learning and working capabilities of these mice. Not all the features could yet be observed in our models, in our condition at least. Like, for example, we did not observe the postnatal growth delay. We also performed some PET analyses to detect in 18 or 23 months old mice if there is any tumor growth. We did not observe it, but we have here a black 6 background, so it's probably not the best prone for this type of diseases. And for the cutaneous abnormalities and the hair and coat, it's a little bit difficult to compare mice and human, but sometimes we had some mice with some wells in the ear, but it was not penetrant. It was only about 1% of the mice. So not sure that this is due to mutation or not. So with this mouse model, where we have reproduced a number of the phenotypic features that I just listed, we have now a new basis, but there is still a lot to do to better understand what are the underlying mechanisms, what is the pathway of the HRAS mutation, how does it lead to the muscular defects, to the hypertension, to the learning and memory defects, and with the ultimate goal to not only to better understand, but also to propose some treatments for these children. And finally, I would like to thank all my collaborators at the Mouse Clinical Institute, Guillaume Pavlovic and his team for the generation of the model, sorry, Patrick Rayleigh and the phenotyping teams, Abdallah Yadi for the breedings and setup of the colonies, and also our collaborators at Bordeaux, who are mainly doing the in vitro analysis. And in particular, I would like to thank the French Costello Association, as well as the National Agency for Research. And thanks for your attention.
Thank you. Nice.
I have a question. I'm curious whether the phenotypes that you don't see in the mouse model have like variable penetrance or expressivity in the human patients, suggesting that they're influenced by modifiers.
So this is something that we are still evaluating, and we have some clinicians in collaboration at Bordeaux. And for example, for the tumors, it's only 50% of the patients that develop tumors. And we analyzed here, I think that it was for the tumor and the PET assay at 18, 23 months of age, we only had 12 mice. So 15% of 12, we had 0. So, but as I said, I think that the black 6 background is probably not the best. So this is something that we have to think about, and how can we try to reveal this tumor progression. And similarly, for the cardiomyopathies, it's not a 100% penetrance in the clinic, in the patients. We have good results here in the mice, but most of the results, also there are some assays where we have a dose-dependent response, and in homozygous mice we have a greater phenotype than in heterozygous mice. Patients are heterozygous. So there are still a number of unknowns, and this is the really the molecular mechanisms that is underlying these phenotypes that we would like to better understand.
Any other questions? No? Excellent. Thank you
Thank
for a
you.
wonderful talk.